Hi, Dave Soriano, and I'd like to give you a brief presentation here on some of the chemical ecology work that I'm conducting with the common name Fiddler crabs, sand crabs, and I have a research program which is involved with detection of odor, sensory chemicals, attractants, repellents with the American cockroach, and now the fiddler crabs, and later this year crayfish. Now the fiddler crab is pretty easy to work with, very active during the day. The male has a very large claw. The life form has been very, very well investigated by life scientists, and I want to see if the fiddler crabs will ingest, metabolize so-called proteinoids, thermal polypeptides, and particularly those that are enriched in the amino acid tyrosine, which is the amino acid which leads to the biosynthesis of melanin pigment, coloration in skin. The fiddler crabs I'm working with have a pretty large concentration spread throughout, dispersed throughout the carapace of melanin, as you'll see in this short video. Here are two of the fiddler crabs. We have a webcam set up so you can follow the work 24/7. If you go to Ustream, letter U, stream.com, you can find my work at Dave, D-A-V-E, Soriano, S-O-R-I-A-N-O, 56. Dave Soriano 56. We have a live cam set up in the campus greenhouse. I'm with the University of Pittsburgh's Bradford campus in western Pennsylvania, USA. And we have Wi-Fi set up there. I'm using a laptop right now to capture live footage. We'll be switching over to a Raspberry Pi in a few weeks. Takes up less room, uses less power. And right now I'm setting up the, let me call it the crab aquarium. And here we see two males that are attempting to establish territory. Another photo as they butt heads with their claws. Small claws used for digging and feeding, and a large claw for display to females and for establishing territory, warding off potential invaders, trespassers. Now the proteinoids. You will find reference in the late 1950s to the late biochemist Sidney Fox, where he was investigating prebiotic chemistry and carried out in the melt phase at 170, 190 degrees, later on below 100 degrees with phosphoric acid, but he was able to make what he called proteinoids. A lot of people refer to them now as simply thermal polypeptides. And you generally have to have about a significant amount of glutamic acid or lysine, particularly the glutamic acid, which melts, forms a cyclic lactam, and solubilizes the other amino acids that you may elect to incorporate into the growing chain. I'll be conducting the synthesis of these thermal polypeptides towards the low-end molecular weight, 2,000 to 5,000 Dalton units, via sulfonane solvent. You can find reference to this in the U.S. patent literature. And what I intend to do is to enrich the polypeptide made this way in the amino acid tyrosine, which is a starting point for the biosynthesis of melanin. Also, with the temperature used, 170 to 190 degrees for 3-4 hours, if you start with the L-amino acid, the one that's found in all proteins biosynthesized from life forms on Earth, no doubt racemization will occur, And I will also start not only with the L-tyrosine, but down the road I will do the synthesis with the D as well. But I want to enrich in tyrosine, and I want to be able to see, do the fiddler crabs ingest the material? Will they ingest it? Will they metabolize it? We will analyze the water. They live in brackish water. Their excrement will probably wash into that water, and we'll be able to see if there's evidence of the thermal polypeptide showing up there. And we will look at the carapace, the exoskeleton, and see the melanin distribution if they do ingest and metabolize the enriched material with tyrosine. And we'll see the effect, if any, on melanin versus controls. This is what we have planned. I have a colleague who's a microbiologist who's also working with life forms, microbes, and he's quite interested in melanin biosynthesis. So he'll be of great help as a colleague on this project. Here's the setup: a 40-gallon aquarium, sand angled down to a basin of brackish water. I use 90% distilled water, 10% of ocean water. Don't use sodium chloride, and I maintain it 10 to 15% by volume, ocean water to fresh water, pH 8 plus or minus half a unit. And if we take you back to this one, we can show you a little footage. That's a male in the corner. There is a tripod that I have actually in the terrarium where I hook up a $15 webcam. Hey. I feed them shrimp pellets. It's a male. You can see the claw, large claw. See the melanin, the black pigmentation on the carapace, as well as the rest of the body. Here's their pool of water. There's an impressive looking male. One in the water and one up on top near the rock. I simply put the shrimp pellets on that large rock, or distribute a few in the sand. The females have two smaller hands, and the male will use the smaller hand for scooping food up. And the ingestion process is fascinating. There's a female in the corner. They move sideways. It's a decapod. There's one. See, he's eating. Small hand used to bring the food to the oral cavity. Claws used for courtship, attracting females, and for threatening other males that are infringing on territory. I'll change the water once a week. These came in from Carolina Biological Supply. I think there's 11 in here. I have another one in a separate tank. As I said, we have a webcam set up. I have a red incandescent light bulb set up so you can view at night. They're rather quiet at night, but occasionally you will see one moving around. So you can log in 24/7 to see what's going on. Here's set up video number 2. Close up of that pretty impressive male. Don't bother trying to get out. I have it boarded on the top with plenty of holes for ventilation. Now, I bet you that claw, I read somewhere, it might be 30–50% of the body mass. If it loses a claw, it'll regenerate one, but it'll grow up on the opposite hand when molting occurs. Half the battle with this webcam is it's a little portable cheapo. There we go. Try to log in on these, zero in on the. Must be very difficult for the animal. You can see the eye stalks. And they're looking for there he goes, he's eating. And they will get that sand very efficiently away from the food. Very, very popular animal with life scientists, ecologists. You can see the melanin, the dark coloration they all have. I'll see how that changes with the proteinoid. I'll offer the shrimp, but I will also have a separate area with the proteinoid and see if they accept it. And then, as I said, we'll analyze the water, because when they bathe, when they jump in that water, there's some of the excrement will go into the water. We'll be able to see if the thermal polypeptide is showing up, and I can measure the mass of the amount I'm putting in the crab aquarium, so on a small dish, and then I can take it out, weigh it, and see how much has been taken up, if any. It'll be interesting in the next few days to see if the coloration changes. Can be hormonal controlled diurnal. If you want to get a hold of me, Dave Soriano. And we have another one set up at bamboozer.com, a webcam. Bamboozer.com, BAMBUSER.com. And there you will find me with D.S.Soriano. And you can see an overhead webcam of a male that I have in isolation right now. Thanks for watching.